Assalamu alaikum class. I'm Saida Mahina. Today we will discuss about BLAST. BLAST is basic local alignment tool. So we'll focus on BLAST now. For that, let me open Google. How are you all? I hope you all are fine and doing well. So for BLAST, we have to search BLAST. And then press the option. BLAST is basically related to NCBI. So you can see BLAST local alignment search tool. BLAST finds region of similarity between biological sequences. The program compares nucleotides or protein sequences to sequence databases and calculates the statistical significance. Let me increase the volume for all of you so you can hear me well. Now, here is nucleotide blast. Nucleotide blast is a tab blast in which we can put nucleotide sequence and we get the pairwise sequence alignment of nucleotides. Here is blast X. Here we paste the sequence of nucleotides and get the pairwise sequence alignment of protein. Now in T blast and we have to put the sequence of protein, I mean amino acid sequence, then we will get the nucleotide blast sequence alignment. So here is protein blast in which we just paste the protein sequence and get the protein pairwise alignment. For nucleotide blast, we have to click on nucleotide blast. And here, let me search NCBI. NCBI is National Center for Biotechnology Information. The National Center of Biotechnology Information is part of United States National Library of Medicine, a branch of National Institute of Health. It is approved and funded by government of United States. So here we need a amino acid sequence. So we can click on in NCBI, you can see all the databases. Here is an assembly database, biocollections, bioproject, biosample, biosystem, books, or CleanWar, conserved domains, dbGAP, dbWAR, genes, and genome. We need a amino acid sequences, so we can click on gene, and we also might click on genome. So C9 or F72. This is a gene. You can search any gene of interest which do you want to do pairwise alignment. Okay, here you can see we have C9RF sequence and with its complex subunit. So for faster sequence, we have to go down, and here you can see FASTA. FASTA sequence is basically text-based sequence. So here you can see, you can paste just accession number or FASTA sequence, or you can choose a file. So here you can just write the accession number. Now you can see NC000009.12. This is the accession number of C9 or F72 gene. But we will paste its sequence. So I'm going to copy the sequence, then I will paste this sequence on blast, our basic local alignment, pairwise local alignment. Basically, blast is used for pairwise alignment. If you want to do multiple sequence alignment, you have to choose some other tools for multiple sequence alignment. So now I'll paste this sequence here, and now you can see here is database nucleotide collection. Here you can see NRNT. NRNT means non-redundant nucleotides. So we will keep it as it is. And now here you can see the program selection. We want high similar sequence, so we will keep it as it is. Now we do blast. First, here you can see algorithm patterns. Click on algorithm patterns. You can see maximum targets sequence should be 100, and expected threshold must be 0.05, and word size 28, and matches and mismatches score in between 1 to 2. Here you can see the gap cost linear, it must be linear, and low complex region and mark as lookup. And here you can see something. Here we can select the organism. I want to check it, this gene in Homo sapien. Here you can see Homo sapiens. Yep. Homo sapiens. Okay. I selected Homo sapien. Now I'll do blast this sequence. Now click on blast, and we have to wait a little bit just to get our result. Today I'll teach you all what is nucleotide blast. In 2 to 3 sessions, we will learn completely about blast, or then we will do nucleotide blast, protein blast, and blast, and all the blast. Here you can see we perform blast, and it means nucleotide blast. We have to wait little bit for our results. If you are all are comfortable with this class, you can subscribe this channel and like the video and comment. If you have any query related to bioinformatics, I'll surely give you an answer. Now here we have our result. Yes, here you can see our limited record include Homo sapien, because we just click on Homo sapiens. Homo sapiens are human scientific name, and we perform blast and we select non-nucleotide database, and here the query length is this, and distance tree. We can click on this also to check an MSA, multiple sequence alignment. So here you can see we can go to GenBank graphics, distance tree of results, and MSA viewers. This is new, so here they put a tag of new in red. Now we can see the maximum score is 50453, and total score is 51473, and query coverage is 100%. E-value is hit by chance, so it is 0.0. The e-value must be 0 or near to 0, it will consider good. And percentage identity is 100%. Accession length is 45227, and this is the accession number. So we'll click on this. First, we have to see its graphic summary. Here you can see the scores are not that much good as I considered it to be. Here you can see if your alignment scores are greater than 40, then if your alignment scores are okay, sorry for the noise. Here you can see we have good score. We have relatively good score. Now you can see if you have alignment score less than 40, it will considered as bad score. And if it is 40 to 50, or the best score is this if the alignment is in red color. This is Homo sapiens subunit, or it gives us all the complete information. Here we can go and check the alignment. This is the alignment which gives us the best score. For that we have to go back. Yes. Yeah. And now I hope you all are understanding it well. Now let me see. Okay. This is pairwise alignment. Now you can see. Here you can see C is aligned with C, C is aligned with C. This is the query and this is the subject. Very well done. The sequence was little bit longer. That's why we get this type of result. From here you can click on the faster and see the results. Here you can see multiple sequence alignment. NCPI provides so much help to analyze the data related to bioinformatics. Here I have some network issue, so we should go back, and now we can check again the graphic summary. Here hover to see the title and we click to show alignments. Okay, they have selected 100 sequences, as you can see, but there are so many gaps, and here you can click to have help. The graphic is an overview of database sequences aligned to the query sequence. These are represented horizontally bar color coded by score and showing the extent of the alignment on the query sequence. Separate alignment region on the same database sequence are connected by the thin gray line. Then mousing over an alignment shows database sequence. Click an alignment, display a box with more detail about the alignment. Link to the sequence alignment itself in the alignment section of this reports. Thank you. So I hope you all know how to do blast and get the sequence and how to analyze nucleotide blast. Then for next class, we will search blast X. Then we will perform T blast and protein blast. Okay, class. I hope you all are fine. Take care. Allah Hafiz. Let me end the class. Okay, class. Now you can have good time with your family and friends. I hope you learned something and see you in the next class. Allah Hafiz.